Previously, we focused on polyclonal antibodies. Now let's discuss monoclonal antibodies. So what exactly is a monoclonal antibody? So let's imagine a collection of plasma cells, and all these plasma cells are identical. Now, if we have some type of infectious agent, some type of antigen inside the body, then all these identical plasma cells will begin to produce identical antibodies for that specific antigen. And all these identical antibodies will bind to a specific region, a specific epitope on that antigen. And this collection of identical antibodies that bind to a single epitope on that antigen are known as monoclonal antibodies. And this is in contrast to polyclonal antibodies because, as we said, polyclonal antibodies are produced by different types of plasma cells. So we produce a different collection of these different types of antibodies. And even though these different antibodies are for a specific antigen, they can bind to different epitopes on that antigen. So this is the difference between monoclonal and polyclonal antibodies. So let's take a look at the following diagram that describes the binding process between monoclonal antibodies and antigens. So we have 1, 2, 3, 4, 5, 6 of these specific identical antigens, and we have 1, 2, 3 different epitopes on each one of these antigens. We have the orange one, we have the brown one, and we have the pink one. Now, this is our collection of 3 identical monoclonal antibodies which were built by the same type of plasma cell. And so what that means is they will all bind to the same exact epitope on that antigen. So let's suppose the orange epitope. So that means once they form that antibody-antigen complex, all these antigen binding sites on the 3 monoclonal antibodies will bind to these orange epitopes as shown in the following diagram. Now, to summarize the difference between monoclonal and polyclonal antibodies, let's take a look at the following diagram. So we have monoclonal antibodies and polyclonal. Now, mono simply means we have a collection of identical plasma cells, so a single type of plasma cell, and they produce identical antibodies which bind to an identical epitope found on that specific antigen. But when it comes to polyclonal antibodies, poly means we have a collection of different types of plasma cells, and so they produce these different types of antibodies for that specific antigen, and they can bind to all different types of epitopes found on that specific antigen. So this is the main difference between monoclonal and polyclonal antibodies. Now, the question is, how can we mass produce our monoclonal antibody? So suppose we want to study a specific type of antibody that binds to a specific type of antigen. To actually study that antibody, the monoclonal antibody, we have to mass produce that antibody. The question is, how do we mass produce it to study it? Well, inside our body and inside the bodies of different organisms, we have many different types of plasma cells. And each one of the plasma cell produces a specific type of antibody that binds to a specific antigen. Now, theoretically, to basically mass produce a specific type of antibody, what we would have to do is take out the different types of plasma cells from our body or from the body of some other organism, and then out of that collection of plasma cells, isolate those plasma cells that we actually want to study because those plasma cells produce the antibody of interest. Now, once we isolate those specific plasma cells, we can then divide those plasma cells many times via mitosis, and those plasma cells, as they grow, they produce those antibodies that we want to study. So in this way, theoretically, we should be able to actually build as many specific antibodies as we'd like. Now, the problem with the method is, as soon as we take out that plasma cell from that organism, that plasma cell essentially dies. So the question is, how can we mass produce the monoclonal antibody specific for some antigen if the antigen producing cells, our plasma cells, quickly die as soon as we take them out of that organism. So the trick is to basically somehow take the plasma cell that we isolated for that specific antibody and turn it into a cancer cell. Because remember, cancer cells are immortal cells. They essentially divide outside that organism. They can divide on any medium that contains nutrients, and they divide indefinitely. And that can be a very useful technique if actually used correctly. So the solution lies in using immortal plasma cells derived from a type of cancer known as multiple myeloma. So multiple myeloma is a specific type of cancer cell of plasma cells. So let's take a look at the following 3 steps in our procedure to mass produce these specific monoclonal antibodies. So in step 1, what we basically want to do is we want to find an organism with a healthy and functional immune system. So for example, let's take a mouse. And what we want to do is, since we want to produce a specific type of antibody, what we're going to do is we're going to inject that mouse with an antigen that binds to that antibody that we want to study. And so if we inject that antigen into the bloodstream of that mouse, the immune system of that mouse will begin to produce the plasma cells that produce the antibodies that bind to that antigen. So after time, when this mouse has those plasma cells following injection, we can basically draw blood, let's say from the spleen, and if we draw the spleen cells, because in the spleen cells we have a high concentration of these plasma cells, some of these plasma cells in the spleen will basically contain the ability to produce that specific type of antibody. So we inject the antigen into our mouse and we wait some time until that mouse produces our plasma cells that produce the antibody of choice. And so then we essentially draw some blood from our spleen, from the spleen of that mouse, and we extract the spleen cells. Now, the majority of these spleen cells will not have the ability to actually build that antibody of choice, but some of these will be the plasma cells that produce the antibody of choice. And so as we'll see in step 3, we're going to want to screen these different types of plasma cells, and we want to find those plasma cells that we want to, that basically produce that particular antibody of interest. Now, in the second step, what we want to do is now we want to transform them into cancer cells. And the way that we transform these normal spleen cells, healthy spleen cells, into cancer cells is by mixing them with the multiple myeloma cancer cells. So if we mix these orange cancer cells with these normal purple cancer cells, the 2 types of plasma cells will essentially fuse in a solution of polyethylene glycol. And once they fuse, we essentially transform these healthy plasma cells in purple into these cancerous plasma cells known as hybridoma cells. So hybridoma cells are hybrid cells between these healthy plasma cells and these cancerous plasma cells. So now in the beaker, some of these cells are basically cancer cells that are plasma cells that produce the antibody that we want to study. And so in the next step, in step 3, we want to take these hybridoma cells and we want to screen them many times with different types of assays to basically determine and isolate those plasma cells that produce that particular type of antibody that we want to study. So we remove all those cancer plasma cells that we don't want to study, and we only keep the plasma cells that produce that monoclonal antibody that we essentially want to study. And so now that we have these cancer cells that are plasma cells that produce the monoclonal antibodies, we can place them on a medium and we can basically grow them indefinitely. And as a result, we can mass produce that antibody of choice. So that means we can either mass produce and study those antibodies, or we can take them and inject them back into the mouse to basically study how the cancer actually progresses and how we can cure the cancer. And we can also actually freeze them, because by freezing the cancer cells, that doesn't actually kill the cancer cells. Once we unfreeze them, they will continue to divide indefinitely. So we see that these hybridoma cells are very convenient because they give us this procedure to basically create any type of monoclonal antibody that we actually want. And this has many, many applications, especially in medicine. And as we study biochemistry, we're going to come back to this type of method because it actually is used in many different areas in biochemistry.